Chapter 6, Growth of Microbial Populations. The learning objectives for this topic are: describe logarithmic growth, explain what is meant by the generation time of bacteria, draw and label a bacterial growth curve, describe what occurs at each phase of a population's growth, explain how a chemostat can maintain a microbial culture in a continuous phase, contrast direct and indirect methods of measuring bacterial reproduction. The topics that we're going to be covering under this topic are mathematical considerations and population growth, generation time, phases of microbial population growth, continuous culture in a chemostat, and measuring microbial reproduction. Growth of microbial populations always deals with logarithmic / exponential growth. One cell will divide and produce 2 cells. Each of those 2 cells will divide and produce 4 cells. Each of those 4 cells will divide and produce 8 cells. In other words, each time you have a division of cells in the population, you end up with twice as many organisms. And you end up with a logarithmic growth curve. We're going to take a look at that when we actually look at a growth curve. Generation time is the time required for a bacterial to grow and divide once. Now, a lot of things go into affecting this generation time. One of them is food availability. They've got to be at optimum temperature to have the shortest generation time for that species. If they have a lower temperature, of course the generation time is going to be longer. And if there are any inhibiting chemicals, that's also going to slow down the time required for a bacterial to divide and grow once. Also depends upon the species. Some microbes, some bacteria just grow faster than others. Eukaryotic cells have a longer generation time than bacteria. That's why fungi oftentimes in lab cultures get overgrown by the bacteria. So the shortest generation time that I'm aware of is 20 minutes, and that's for a prokaryotic cell. Whereas there are other organisms that take days. I used to do testing using an acid-fast bacterium that had a generation time of about 8 hours. And it took 21 days before you could see colonies. Boy, you didn't want to mess that test up. Hoo-wee! So let's look at the phases of microbial growth, or a growth curve. When we take inocula and put it into new media, oftentimes there is a lag phase, or a time when there's no increase in cells. They're not dying, but they're not dividing. The lag phase is when the bacteria are assessing their new environment and turning on or turning off genes as may be needed. So suppose you've been growing them in an all-protein media and you switch them over to lactose, and they're able to use lactose. They're going to say, hey, there's lactose, turn on the lactose genes. And so it takes time for them to turn on those genes, transcribe the mRNAs into proteins that can actually use the lactose. Then the organism can use the energy from the lactose catabolism, and they start to divide. It may be that they were in a stressed condition, low nutrients, other stressors like high temperature. It takes time for them to recover from being in a low metabolic state to turning on genes to produce more enzymes, etc. So that's the lag phase. The log phase, or the exponential phase, is where they are growing just as fast as they possibly can. They grow, they divide as fast as they can. During the log phase, most bacteria are at their most susceptible to antibiotics. Also, that's when you want to do your gram stain. Later on, we get some weirdness in the cell walls, and you may get some funky gram stain results. As food decreases and waste products start to develop, remember we're talking either on a plate or in a tube, you enter what is called the stationary phase. This is where you start to have equal numbers of cells dying as cells being produced by division. So unlike the lag phase, where you don't have cells dying, or the log phase where you have more cells dividing than are dying, you have an equal number of each, and your numbers plateau. If you don't add more food, take out waste, the culture will enter a death or decline phase, where you have more dying cells than you have cells being produced by division. Now, some cells will go into a slower metabolic rate so that they can survive the bad conditions. Some bacilli are able to produce endospores, but in any case, you've got a decrease in your cell numbers. So, you may be asking yourself, how do we tell what the cell numbers are at each of these phases? You can't see them, they're too small. Well, I'm glad you asked, because now we get into how we measure microbial growth. Let's talk about direct methods first. The first one is viable plate counts. That's basically where you count the number of living cells. So, how do you do that? By either a pour plate or a spread plate, like what we talked about in the last topic. Another method is membrane filtration. You use that when you have a high volume of fluid and you're expecting a low number of cells. I use this a lot when I was testing bottled water samples and various other water samples. So basically what you do is you set up a membrane filtration system where you have a sterile funnel, you've got a membrane here, you've got a filter here, and the fluid goes into a flask and you have a vacuum on it. So the idea being that you get rid of the volume, you trap the microbes on this membrane filter that is sitting on top of this glass filter. You remove your membrane filter and you put it on media to feed them, and you get colonies growing. And it looks like this, especially if you put in an indicator that turns the colonies this lovely blue color. Another direct method would be direct microscopic counts. This is where you're taking a look at the individual cells, and of course you have to use the microscope. Now, we also use two different types of equipment that help us keep track of these little guys. So a hemocytometer is used in counting blood cells, and you have this little glass slide that has overflow troughs, and there's enough space here for a known amount of fluid, and then you put a cover slip on, and it has a grid. Now this one, the Petrov-Hauser, is generally used for bacteria, unless you work in my labs, and then we use just a hemocytometer. But here's a nice blow up of the grids. It makes it easier to count those little dots if you have a grid. So I can count this square, and go on. And because I know exactly how much fluid is under here, I can use that information to determine how many cells per mil I've got. And then, if you're lucky, you have electronic counters which do all of this counting for you. One of them is a Coulter counter, and another one is flow cytometry. Now, with both microscopic counts and electronic counters, you can add stains that can tell you whether a cell is viable, alive, or dead. Another direct method is called the most probable number method. This is used by a lot of water departments because it's quicker than viable plate counts, and it's also differential most of the time. So what you do is you take your water sample from the local water supply and you do dilution. So you get an undiluted sample, dilute it 1 to 10, 1 to 10, giving you a 1 to 100. Then you take 1 ml and you put them into tubes of broth that is very specific for the organism that you're looking for, and it has a pH indicator to indicate when certain carbohydrates are fermented. Then if you have any cells at all in the tube, turns the indicator yellow, indicating fermentation. So, for your undiluted, I had 4 positive tubes. For my 1 to 10 dilution, I had 2 positive tubes. And for my 1 to 100, I had 1 positive tube. Then I would go to a chart that had all of the information from years of testing this kind of test to give me a most probable number of bacteria per 100 mils. So I'm going to go down to the 4s, then I need to go 4 to 2, and then now I've got 4 to 1, so I'm down here right at the bottom. So I have 26 bacteria per 100 mils. So that's the most probable number. Now let's talk about some indirect methods. In other words, we're not looking at actual numbers of viable cells, actual number of cells. Instead, we may be looking at metabolic activity. That would be, we take a standardized temperature and establish the rate of microbial growth. So the more cells I have, the more metabolic activity I'm going to have. So we can measure pH, we can measure nutrient use, we can measure waste production of something other than organic acids. All of this would be measuring the metabolic activity, which measures the number of cells indirectly. Another method would be dry weight. Some cells just do not lend themselves to viable counts or direct microscopic counts. One of them would be penicillium. If you're growing a mold and then you've got all those hyphae intertwining, it's really hard to follow the hyphae to figure out how many cells you've got. So instead what we do is dry weight. So we take a filter that is dry, we weigh it, then we take a standardized amount of our penicillium culture that's growing penicillium in our factory, and then we filter it, catching the organisms on the filter. We dry it out, weigh it again. That tells us approximately the mass of mold that we have growing in our fermentation chamber, and we can tell whether the numbers are going up or whether they're going down or whether they're at just the right weight. Then there's turbidity. Cells block light. And so when you look at a broth that has a lot of cells, they're blocking enough light that the media is turbid or cloudy. So we use this to measure the growth in broth culture. Now you can either eyeball it, which is what you're probably going to do in lab, or you can use a spectrophotometer to measure the absorbance and you can get an actual number. Turbidity has its limits. You have to get at least 1 million organisms per mil before you start getting visual turbidity. Spectrophotometers can measure a little bit below that, but not much. So you're measuring, now see, I know from experience what the number of cells per mil are because lots of people have measured the turbidity, the absorbance, the ability of the broth culture to block light, and then used direct microscopic count to determine the number of cells in the broth culture. So you've got to have greater than 10 to the 6 cells per mil, or 1 million organisms per mil, to effectively use turbidity. Also, the cells need to be suspended uniformly within the broth. If you have a whole lot at the bottom and not a whole lot at the top, you're not going to get an accurate absorbance reading. So that's it for this topic. Here are the reminders of the topic learning objectives. Remember to write down your questions.